My name is Professor Paul Freemont, and I'm going to tell you a lot, hopefully in the next 35, 40 minutes, of a very exciting new technology development called synthetic biology. I'm also going to try and convince you that synthetic biology is going to be one of the cornerstones of the new way that we do things, and that will lead to a very growing economy, which we call the bioeconomy. But first of all, we're just going to start off, and I'm going to sort of tell you a few little anecdotes and sort of get you in the mood. So my first one is what if? So what if, instead of building buildings, we planted them as programmable seeds? What if we engineered trees or plants that could somehow capture sunlight during the day, but at night emit that light out as some sort of bioluminescence? What if we could cover whole buildings in engineered photosynthetic algae that could absorb carbon dioxide, which is a huge problem, particularly in London at the moment, to produce heat and also to produce biomass? And that's Hong Kong, by the way. What if we could make clothes or even make water filters out of bacterial cellulose? What if we could take living organisms, re-engineer them, and implant them into your body so that they could detect all of the metabolic things going on in your body, and if they detect something that's not quite right, deliver something that could alter your metabolism to make you feel better, called living therapies? What if we could take that a little bit step further and make a probiotic drink, like a yogurt drink? And a lot of people drink these kind of drinks, but we're going to engineer the bacteria in that yogurt, and we're going to make them sense things in your stomach or in your gut and in your colon, such that when you go to the toilet in the morning or whenever, and you look at your feces, if it's a particular color, you think, oh dear, I need to go to the doctor because I've got some kind of problem going on. Okay, what if we could engineer bacteria to target cancer cells in your body and actually deliver killing signals to those cancer cells? What if we could engineer naturally occurring bacteriophage? These are the organisms that kill naturally bacteria, but let's re-engineer them. Let's make them really specific for pathogenic bacteria. What if we could take your T cells, re-engineer them, put them back in your body so that they can then target cancer cells in your own body? What if we could take your skin cells and maybe just let's sort of put them back to their embryonic state and then reprogram them so that they can make new organs for you? So your skin could produce a new liver, a new kidney, whatever you need depending on how you're aging. What if we could take little pregnancy strips and go around and test for viruses or for bacteria or parasites, just like you do like a litmus test, and you could pull it out, get a color, and suddenly you would think, oh dear, there's a very nasty virus around, or whatever. What if you could take all this landfill rubbish that we've got here and convert it into, I'm not sure why it's gone off the screen, but convert it to, could you get it back on? Convert it to recyclable bioplastic objects. So believe it or not, we push all of this stuff into the land. We bury it. I remind you, this is 2016. This is 2016, and we take all that stuff and we put it in the ground and we just cover it up. So that just does not make sense. What if we could take things like petroleum, oil, gas? We know that these are limited resources. We know they have environmentally unfriendly properties. But unfortunately, these commodities produce all of the chemical building blocks that we need for everything that we take for granted. Everything you've got on you now will have been produced or manufactured from chemical building blocks that will come from petroleum or from coal or from some of these petrochemical sources to make all of these products. But we really need to move away from that. So what if we just replace that with biology? And let's use synthetic biology as a mechanism for making all of these commodity chemicals in a much more environmentally friendly way. What if we could reprogram cells? This is at the heart of it. Let's reprogram cells, either bacteria, yeast, small molecules, and let's make them little mini factories and so that they can manufacture and produce all the things that we want and all the things we need, and essentially we just grow them up like we do beer. We just have massive fermentation factories that are producing all the products that we need. And finally, you've arrived at the world of synthetic biology, because all of those what-ifs are pretty much active research programs ongoing, not just at Imperial College, but around the world. So they're not fantasy. These are real realities that are happening now. So what is synthetic biology and why now? So everyone gets very heated about this subject, and I'm going to try and take you through now for a few minutes what is synthetic biology, why is it so important, and what's different about it. So there's the formal definition, okay? So synthetic biology aims to design and engineer biologically based parts, devices, and systems, as well as redesigning existing systems. And there are a whole bunch of reports written on synthetic biology around the world, including the UK. But if you want to put it simply, synthetic biology wants to use engineering principles for the design, construction, and reconstruction of biology, but we want to do it at the genetic level, and that will become clearer in a minute. Or even more simply, synthetic biology is actually engineering biology. So let's think about life. We need to sort of, take a step back and say, well, what are living systems? So if we look at living systems, they are self-organized. They interact with their environment. The systems are maintained by flows of information, by energy, and by matter. I mean, living systems are truly extraordinary. And the code that runs, if you like, living systems on Earth is encrypted by this iconic molecule, which I'm sure everyone in the room recognizes as DNA. So let's think about DNA. So what is DNA? What does it do? So DNA is like an operating system. So we just consider a living system as a computer or a mobile phone or whatever. There is an operating system in life that essentially runs that cell, runs that organism. And DNA is essentially the code, is essentially the operating system for living systems. Now, just to drill a bit further, it's a very simple molecule. It's iconic and simple. It's got 4 building blocks shown on the left there. The names are not important for this particular point, but they are GCAT, if you like, as an abbreviation. They pair up, so G's or with C's, A's or with the T's, and they form this beautiful double helix here. And what's really important, I think, for the context of synthetic biology is that the information density of DNA is ginormous. So 1 million bases of DNA sequence information, and we normally write it as a sort of string of letters, GCAT, ATG, blah, 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 is about a megabyte of information. So you have 3 million bases in each of your cells, which means you have 3 megabytes of information in each of your cells, and you have billions of cells. So the information density of DNA is extraordinary. If you unwrap all the DNA you have in all your cells, you can reach the moon and back 600 times. That's an extraordinary molecule. Compact, organized, beautiful, at the essence of all living systems. So, how is this code translated? I told you it's a code. What is the information, what does it store, and how is it translated? So the execution, if you like, of the genetic code is through 2 processes, biochemical processes, called transcription and translation. The details are not important. The point is, it is translated. It goes through an intermediary molecule called RNA, and it produces something called proteins. So what are proteins? Well, proteins are the workhorses of life. So the simple relationship is that the code in a living system codes for proteins, and I've shown you a whole array of different protein structures and functions. And proteins self-assemble. They can form large macromolecular machines. They carry out all the complex chemical transformations in your cell. They form dynamical structures because cells have shape and they change and alter. They sense and respond to the environment, and they perform complex signal processing. Proteins are it, as far as I'm concerned. They are really fantastic, incredible. So DNA codes for proteins. So where did all this come from? So basically, we've got to go back only about 60 years to this molecular biology revolution that was driven through the extraordinary discovery and structure of DNA. We then learnt that we could clone DNA. We could move DNA around between organisms. We take one bit of DNA from one organism and put it in another. And that was called cloning in 1971. We then learned how to read the code, called sequencing, 1977. And then finally, we learned how to chemically synthesize it, actually in a test tube, to make the code. So we learned how to move, read, and write DNA in a period of, in terms of the history of humanity, extraordinarily small time. And what that resulted in was what we call the the genome revolution, which we are well and truly in, culminating in 2001 with the publication of our own genetic code, if you like, the human genome. But all of these other organisms on the planet, we now have their genetic codes. We have their instruction sets. We don't necessarily know how they all work, but we have the basic instruction codes. So why is this? Well, like everything, economics drives everything. The cost of reading DNA is shown on this blue little graph. You can see that it is phenomenally cheap to read DNA now, in the sense that you could have your whole genome sequence for a very small sum of money. And in fact, you can go and do that. Now, writing DNA, i.e. synthesizing it, is not quite at the same stage as reading it. But it is coming down. This is a logarithmic scale, by the way. And there are clear indications that the technology is reaching a point where there will be a tipping point when it will be as cheap to write DNA as it is to read DNA. So synthetic biology has emerged at this point as an interdisciplinary science and engineering field, because it's suddenly realizing, well, maybe we can treat this DNA program, which I've described to you, as a program. It's programmable. Can we reprogram cells? Can we actually treat it like an algorithm, if you like? However, biology is complex. I'm not going to go through all of the complexities of biological systems, so this is where the engineering comes to play. So in order to deal with this huge complexity of biology, not only in scale, complexity of biochemistry, complexity of time, living, dynamics, etc., evolution, whatever you want, adaptation, we are going to bring something like engineering as a discipline to bear on biology and use things like systematic design. Now, the reason for doing that is because engineers, as you know, have been using computer design, standard parts, modules, devices to build and test things that are all around us. Everything in the world that we take for granted has gone through some kind of engineering process, and engineers really make the world what it is, actually. And stability and robustness of engineered systems are absolutely key. So the question is, can we apply those same principles of engineering to biology? So this is where synthetic biology comes in. So synthetic biology wants to provide a framework for engineering biology. And it wants to consider the parts of biology as pieces of DNA, which I described. And there are this kind of what they call an abstraction hierarchy. Don't need to go any further with that, but basically what it's trying to consider is you have parts of DNA which could become standardized, and therefore you could start building biological systems by putting different standard parts and modules together, and then doing computer design and prediction and et cetera. So this is where the field sits at. So synthetic biology is trying to build new biological systems using standardized DNA parts that can be used in different applications. So when we consider that and go through that sort of intellectual kind of journey, if you like, you end up at this context, which is the engineering design cycle. So now we're really bringing to bear all of the principles of engineering design to molecular biology. So we're beginning to say, well, okay, if we want to build a cell that has a particular functionality, let's think about designing it. So what are the natural genetic elements we need? How do we repurpose them? How do we refactor them? How do we go and get genetic parts that exist in big database of registries that have been standardized? How do we build computational tools to allow us to design those new algorithms, those new genetic codes? And then how do we build them? How do we test them? And how do we learn? Now that whole cycle is very, very powerful because that's pretty much underpinned everything that we have done so far with engineering. But we've never really applied it to biology at the genetic level. And that is what synthetic biology is aiming to do and is doing. And the beauty of that is that you end up with a synthetic biology engineering framework, which then can be applied to multiple different applications as shown here. And I will talk to you a bit more about these applications in a while, but they stretch from bioremediation to chemicals, crops, therapies, diagnostics, biomaterials, etc. Now, what are the organisms that we want to engineer? So synthetic biology is engineering organisms that are shown on this screen generally. So these are yeast, algae, bacteria, plants, mammalian cells, and human stem cells. So notice there's a whole variety of different organisms that synthetic biologists are beginning to engineer. So another way of putting it is that synthetic biology wants to apply engineering and wants to engineer cells essentially to make useful stuff. That's a very simplistic way of thinking about it. And the useful stuff is, as I showed you for the what-ifs, the chemical industry, consumer products, pharmaceuticals, everything you can think of that we need as a society, we want to use synthetic biology as the way of making it. So what has this got to do with the bioeconomy? So let's think about the bioeconomy. So what is the bioeconomy? It's a sort of nebulous word, but it's an incredibly important concept. So it's an economic activity derived from bio-based products and processes which contributes to sustainable and resource-efficient solutions to challenge the challenges that we face in food, chemicals, materials, energy, environment, health, being, et cetera, et cetera. So it is actually at the essence of time that we think about how we manufacture and produce things in this kind of biological context. Now, it's not new. We've been exploiting biology. The bioeconomy has been around for 10,000 years since we started domesticating animals and doing crops. And shown here, you can see in ancient Greece, they were making, ancient Egypt, they were making bread. They also learned to make wine quite early on in humanity, which is a good thing, I think. And they certainly learned how to plant crops very early on. So this whole idea of exploiting biology for mankind's use has been around for thousands of years and continues to be. And of course, we now have very modern, efficient technologies that allow us to have whole fields of corn or food or the bulb fields in Holland where I was recently. We also manufacture very complex pharmaceuticals using biology, drinks and food, and so many other things that we use biology. So this is where we are now. And of course, the use of technology has accelerated the exploitation of biology enormously. So here I show you crop yields 1950s, crop yields around the turn of the 2000s, a massive increase in efficiency and productivity of agriculture, for example, is one example of how technology can impact on these kinds of processes. Now, not to become too gloomy on a beautiful Saturday afternoon in South Kensington, but we are at a particularly vulnerable point in the history of humanity, I think, where there seems to be a convergence of different factors that are not really clear how we're going to solve them. So the first factor to note, and you probably all know this, is that the population of the world is growing rapidly. So 9 billion people by 2050 and beyond. The second thing to note on this particular diagram is shown the land mass we need to plant to feed the world. So currently, with crop protection, we're down. This is the total land mass of the planet, shown here, including deserts and places that we cannot grow crops on. This is the current use with crop protection. If we had no crop protection, it would be that. And this is the predicted estimate that we would need we would need in 2025 to feed a world population of 8 billion without crop protection. And that's about 5.9 billion hectares. Now, the land, total theoretical land on the Earth is 13 billion hectares. So by 2025, if we didn't use crop protection and any of the interventions that people seem to be very against, we're going to end up using half of the planet to grow food, which seems an extraordinary situation to be in. The other little point, I think, which everyone's aware of, is the increasing concentration of carbon dioxide, shown here, this graph, which I think is a respectable set of data. There are many data sets out there. This is one I picked, which looks to be quite accepted. And of course, the global temperature change. And there seems to be a direct correlation between the carbon dioxide concentration and the global temperature. Now, this leads to other problems. So here I'm showing you water consumption. So now we're looking at water consumption for the world in the top line shown there. And in 2025, we will need 40% more water than the current supply levels if we're going to provide water for all of the world. And then finally, energy. So this is the world's energy consumption by fuel. You'll notice here at the top, there is a little bit of the percentage shown as renewables and shown as other types of renewables like wind and hydro, but it is a very, very small percentage. And you will notice also shown in this graph that oil, petroleum, natural gas, and nuclear, with quite a significant bit, are still going to be absolute key parts of energy requirements for the world, predicted anyway. Now, of course, that could change. It's just a prediction. But I think when you put all that together, so we have food, we have water, and we have energy, key components of living systems, and we have alterations to our climate that we're uncertain about, we have a problem. And you don't need to be a rocket scientist to go on Google and find that the Earth is under pressure. I mean, these are just some examples of the pressure the Earth is under, but it is under a big pressure. So I want to now go back to the bioeconomy. Now, how can the bioeconomy step in to help this whole process? So if we look at the bioeconomy and what it means, if we think about sunlight and farming, forestry and agriculture as one of the base points of the bioeconomy, you can see from this graph that we can then make renewables and raw materials and feedstocks. We can then go from there into the world that I'm going to tell you about, which I told you about, synthetic biology and bioprocessing and making stuff, reduce the dependency on petrochemicals, less waste, any waste can be recycled into byproducts, which can then go back into, by new processes, into renewable waste sources, and any of the nutrients can then go back into the earth through a kind of sustainable, interactive, interconnected economy, if you like. And the beauty of the bioeconomy is that it will essentially reduce our dependence on fossil fuels. It will produce less waste to landfill. It will produce new economic opportunities for technology developments. It will provide some energy security, some jobs and economic growth, and a much more resource-efficient society. So I think at this point in time, the bioeconomy is an extraordinarily important part of where the world needs to look. Now, the value of the US bioeconomy in 2012 was shown here. So it's about 324 billion. Now, having said that, the US economy is worth about 17 trillion, but that's still a very significant proportion. I mean, it's about 2% or something of the US economy on the bioeconomy. But you can see here, when you break that down, that about 66 billion on biochemicals, biologics 16 billion, food and agri 12 billion, and biofuels about 9.7 billion. The industrial bio side is quite big in the US bioeconomy. Now, in the UK bioeconomy, it's estimated to be about 150 billion, which is actually surprisingly about 2.4% of our total economy. We calculate it in a slightly different way, but it's completely legitimate. And we have a very strong technology base that could accelerate the bioeconomy. Now, the UK government has recognized that synthetic biology is one of the 8 great technologies and has put in over £300 million. And it's this interrelationship between the opportunities in the bioeconomy and the engineering of biology that are now coming together to tackle some of these very big problems. So where can synthetic biology impact? Well, I'm just going to be absolutely honest and say, well, to be honest, everywhere. Because what we're talking about is the ability to efficiently accelerate the engineering of biological systems that can then slot into all of the many different opportunities that there is in the bioeconomy for producing chemicals, for producing waste, for reducing waste, for producing energy, for producing heat, whatever. It can really provide solutions and technological solutions for all of these different components, including harnessing sunlight. Now, the World Economic Forum in 2010, I think, said that, or 2012, in terms of the top 10 emerging technologies, synthetic biology and metabolic engineering were number 2 in their list. And if you ally that to the global market of synthetic biology in 2009, it was 5.7 billion. It's estimated to be around 38 billion in 2020. Now, those numbers are all, predictions, but it is a very rapidly growing opportunity. But I'm going to stop there because let's think about it. What I'm saying to you is that we're going to engineer life. We're going to treat life as an engineering matrix. We're going to treat our systems as part of components, bits of DNA that we can put together in different ways. And, you know, there is a big issue here because society has a voice. Not all, I mean, the Daily Mail has a really good voice here if you look at that. And you can see at the top there, the Daily Mail says, scientists accused of playing God after creating artificial life by making designer microbe from scratch, but could it wipe out humanity? And there are some really other good headlines here. Put a stop to the synthetic biology pollution. What else have we got? Alien life form is grown in lab. Scientists are unnatural DNA strands to the genetic code of bacteria to create a new strain. Brewing bad. Scientists find his way to cook up heroin. Now, these were all scientific discoveries that were published where newspapers and NGOs have picked up on and reconfigured the media and the PR about those stories. And I think the one on the ice cream really brings it home. It's, you know, when everyone says that image can produce so much punch, I think this image is extraordinary because you've got a pipette going into lovely ice cream, extreme genetic engineering in your ice cream. And that, you know, that's a really evocative image. And this is all about producing vanilla. Anyway, I think it's fair to say that the limits of synthetic biology or engineering biology, I personally think will not be technical, but they will be societal in that the applications and utility of the technology will need to be proven and accepted. And at Imperial College and around the world, we're developing a framework called Responsible Research Innovation Framework, which I'm happy to tell you about afterwards. But you can see this tension between the global challenges that we're facing, the opportunity that bioeconomy offers, and then the societal acceptability of a new technology which is laying out to engineer life in a much more accelerated and systematic way. So what are we doing at Imperial to accelerate the responsible development of synthetic biology? Well, I'm going to tell you in the last sort of 5 minutes of my talk the acceleration that we've developed, which is we have the UK National Translation and Commercialization Centre for Synthetic Biology called City. We have a mission, which is to translate and commercialize responsibly the technology. We want businesses, we want startups, we want to increase the UK skill base. We want to open a positive dialogue on the technology. We have 17 university partners all around the UK. We have a lot of companies partners, both big multinationals and small, medium enterprises. And we have a whole series of supporting partners. And we have this mission, which is to try and bridge this gap between the wonderful academic research that's going on in the UK in synthetic biology and try and pull it through into address some of these technological challenges and opportunities that we have. And there's just some lists of things that we're doing in entrepreneurship, commercialization, skills training, resources, proof of concept funding. And we've got a very exciting group of small companies and big companies changing their technologies around synthetic biology. Finally, I want to end with one thing, which is the Foundry. That was opened in the 6th of April 2016. So this is the first Foundry in Europe, actually, and probably around the second or third Foundry in the world. So this is a Foundry which brings together automation and systematic processes to enable the design cycle for synthetic biology. So what you're about to watch, I think, is the future. I think it's a new industrial revolution around engineering biology. And I'm going to just sort of play this movie. This is a whole bunch of... Turn the lights on. There's the lights.
伦敦的帝国理工学院实验室里，机械手一直不停地工作。他们组装的不是汽车或者钢材，而是基因，也就是有遗传效应的 DNA。
جديد افتتح في لندن ويتبع جامعة Imperial College. يتخصص هذا المصنع في تجارب الاصطناعة الحمض النووي الريبي المعروف ب-DNA.
This might be the future of British manufacturing. Teams of collaborative robots building not cars or steel, but DNA. This is the Foundry, a £2 million centre which opens today in Imperial College, London. It aims to industrialise the process of preparing DNA for use in synthetic biology. Those DNA building blocks will be used to create biological machines which could create new drugs, vaccines and even fuels.
This is the future about how we're going to build and manufacture, really complex molecules like drugs or pharmaceuticals or even commodity chemicals. The idea of removing our reliance on petrochemicals, on, all synthetic chemistry, and not sure why that crashed, but it crashed because I said something wrong. Like all good things, it worked. But I may try and get it back for you, if I may. That was Sky News, and it was franchised all around the planet. And basically, they did a very nice story on our foundry. If I don't get it back, I'll try one more time, and if it doesn't work, we can bring this talk to a conclusion with some questions. So I will wind it forward, because I would like you to see the actual bit that the reporter did, which was very good. Let's see if this works. Okay, right, that's me. Right.
Synthetic biology involves engineering bits of DNA that aren't found in the natural world. Engineers do this by
Hopefully that will work. Oh, okay, sorry. Oh, I'm screwing this up. Okay, let's go. There we go. Okay, play that.
That aren't found in the natural world. Engineers do this by assembling the molecules that make up DNA into new combinations. That new DNA is inserted into living cells. The cells then become biological factories, making chemicals like pharmaceutical drugs or biofuels. Since the human genome was decoded in 2000, technology has been advancing rapidly, giving us the ability to rewrite DNA down to the individual gene, or even to create living cells from scratch. Synthetic biology is, some hope, the next industrial revolution, one where the machines aren't made of steel or iron, but of living things designed by us from the DNA up. If that's the case, then this is one of the first factories of the new biological age. Across the UK, synthetic biology companies are springing up. At Warwick University, Chloe Quee is developing a new type of peanut using a biotechnology called CRISPR.
We're knocking out the genes that create the proteins responsible for allergic reactions. The great thing about CRISPR is that it's much easier than technologies used in the past. So it's cheaper, it's faster, and it's very, very easy. All you need to do is have this address that tells you which gene you want to target. You design that part, and it's very, very easy.
At a time when more traditional manufacturing industries are in crisis, synthetic biology is one area where the UK is a genuine world leader. It's an industry of the future and one that will hopefully arrive soon. Tom Cheshire, Sky News.
Thank you. If you put the lights up, sorry about the break in the thing. But that, I hope that indicates to you where we're going. And thank you for listening, and if you've got any questions. Yeah.
So when it comes to sort of societal acceptance of it and that being a big challenge, what is the solution to that?
Two-way discussion. To be open and to share what you're doing, to get people's opinions about it, and to think carefully about what the downstream consequences of your technologies could result in. And in some senses, even modify that if there are some negative, unacceptabilities. So I think we're in an era where, with social media, with the media, with everything around us, that we are constantly under the spotlight. And I don't think as scientists, and we are publicly funded. We are funded by the public, and myself, and the public. So in some senses, we have a responsibility to share what we do openly with the public. It's a complicated piece, but it's one I think we will continue to do.
The slide you said, protected land, untouchable. What does that mean?
Protected land, I think I'm not sure I was protected. I think what I was trying to project there was the total amount of land mass on the world, and some of that land cannot be grown, cannot be used for crop growing. I think that's what the context of that was.
So the first on the left is, can you go through the
Yep. And I think the point I was trying to get across was that the technology developments that we've been developing in agri-science for the last N years has resulted in accelerated crop growths, accelerated protection of crops from pests and other types of interventions. And I think if you do the comparison and the prediction of where you have crops without crop protection versus crops with crop protection, and this is just an analysis done by a group of pretty expert people in the United States, those are the kind of things that you arrive at. So the point, I wouldn't take it literally. The point it's trying to make is that the land required to produce the amount of food for the growing population is so significant that we're at a point where we need to think seriously about how we're going to do this.
We're now 7 and maximum will be 9. How can we, why do we need double the land?
That's without crop protection. So the point there, so the first one is crop protection, the second one is without crop protection, the third one is without crop protection.
What is crop?
Well, all the spraying that goes on to stop insects, to increase, to fertilizers. The sort of things that a lot of people get quite upset about. I mean, there are obviously good reasons for that, but there is this ecological balance between preserving the ecosystems and growing food, and this is just illustrating that challenge. It's not making a political statement, it's just saying there is a major challenge there that we have to address.
So with regard to real-world applications, what's the next big thing that we're going to see, do you think?
In terms of real-world applications, there are two things that are coming down the pipe. One is that you've already probably seen it, which is the gene drive application for reducing malaria or Zika virus, which is highly controversial, which is releasing genetically engineered mosquitoes that would go in and essentially create an infertile population of mosquitoes, which would sort of essentially drop that population. That's happening now. It's a company called Oxitec. It's a US company out of Oxford, based at Oxford, bought by US. They're doing field trials in various parts of South America at the moment. That's highly controversial because that is an engineered insect released into the thing. What I think more likely is going to be the manufacturing ability of making chemicals and making biochemicals and making very complex pharmaceuticals. And there are a bunch of those processes that are coming down the pipe. So in terms of real world, I think what we're going to find is there are going to be bits of the chemistry industry which are going to be replaced by biology. And I think most people are thinking they're going to be slot-in economically viable solutions. There's no point in doing a technology if there's not an economic fit for it. And one of the problems with industrial biotechnology at the moment is that it's actually much cheaper to get oil out the ground and make all those commodity chemicals than it is to do it in a kind of slightly more environmentally friendly way. So I think one of the issues that the bioeconomy is trying to address is how do we make the economic drivers work such that the technology can be taken into the real world. The technology is developing and we'll be able to do it, but then the course is how can you implement it? If there's not an economic driver for it, and this is a big challenge. So the question is how close do we get to the edge when we suddenly realize, well, let's pay a bit more for the products that we need, kind of thing.
Sorry, the sort of DNA analogy is sort of code to
Yeah.
program and stuff. How about, do we understand the bug, per se, within those codes?
Do we understand the what? Sorry.
So the bug is, so if there's genetic mutation and stuff like that,
Yeah.
how can we deal with it in a robust way?
Yeah. There are a lot of, I could give a lecture course on, we know a lot about DNA. It's mutatable, it's repairable. One of the big things about engineering biology is that you have to have engineered biological systems that are very robust in an engineering context, but of course DNA and life has adaptability and varies and mutates depending on stress signals and all the rest of it. So that is an engineering challenge that a lot of people are thinking about in synthetic biology. Because clearly if we've got a device or a logic gate or some sort of circuit that we've built and we put it into a cell and it works, for 4 hours and then it stops working because it's being mutated, then that's not very good engineering. But I would consider that an engineering problem, kind of thing.
Can I ask a question about the scalability of
Yeah.
mass chemicals? So if you look at something like those low density polythene, it's pretty easy to set up a plant making 150,000 tons a year. But bioreactors
Absolutely.
are rather different. So could you say if you took a synthetic polymer like PLA?
Yep.
I'm trying to get a scale of how
No, you're spot on. So one of the big challenges, I think, for synthetic biology is for industrial biotechnology is scalability, scale up. And I remember going up to Runcorn when Shell were up there presenting this new technology to some of the Shell guys. And they pointed across to this big biorefinery and it says, well, that's the scale we work at. And I'm thinking, well, and I'm presenting 50-liter, this is a real big challenge. And it's a challenge that we need to address. But I would argue that the scalability issue will have to be through the engineering process. So it'll be actually working with organisms and getting them into an engineerable state that would allow their mass growth under these very gigantic conditions. The other complexity of bioprocessing, which is what you're talking about, is the product production, the chemical engineering process, which is an economic driver. So how easy to get my product out of the cells? If I chemically refine it, I can do all sorts of chemical extractions, and I can have a nice cheap product that's so that's another challenge. How do we get products out of these cells into economically viable? So these are big challenges. I don't have the answer. If I did, I'd be on Bermuda now, probably, or some island somewhere. But I don't have the answers. But those are the challenges. What I'm saying at the technology level, at the R&D level, the new kind of systematic processes for engineering cells is actually happening now. And part of that process will be the upstream challenges that you quite rightly present, which is scalability. But everyone in the field is working desperately hard to try and work at that and try and build those scalability things into the design solution at the very early stage in the R&D level. So it's not a very good answer because I can't say we can do it yet fully, but we can do certain things. So high value, low volume chemicals, complex chiral chemicals, complex pharmaceuticals, complex natural products that are very difficult to chemically synthesize. We're beginning to see those coming through the synthetic biology pipeline. But PLA and all the other huge, commodity chemicals, but the economics will have to alter, or the world will go under. Well, whatever. Okay, I sense everyone's had enough of me, so thank you very much for listening. Anyway, I'm around if anyone wants to talk.